So what genotype of individual does this P0 cross produce? Right there. What did you get? Heterozygous at both loci, so a dihybrid. So you get a big B and a little d. And you get a little b and a big D. And then what sort of nomenclature has to go in there? See slashes between the similar letters. And what does the dot in between the B's and the D's mean? Yeah, unknown, what do you call it? Unknown linkage. Yeah, so we're trying to find out, no, you're totally right. We're trying to figure out whether or not the B and the D genes are linked. So we don't know if they're on the same chromosome or different chromosomes. So we put that dot in between them. This is all review. What genotype do you put in this second blank in the F1 generation? If we're going to, right, if the question is, which it's going to always be for this chapter, genetic mapping, how far apart are B and D, then we need a tester genotype to cross to a dihybrid. So the tester, little b over little b, so contains recessive alleles at both loci. And then we get these four types of genotypes in the offspring. And the numbers that are given here. So these are the numbers of different individuals with different genotypes. And we know these because we can actually collect the F2 offspring from the cross. We take their DNA and we ask at these two genes, which versions of the genes do they have? Big capital letters or lowercase letters. So we experimentally determine those numbers, 69, 81, 422, 428. That's why geneticists need a lot of help from students doing research, because we need a lot of people to count and genotype a lot of organisms to figure out whether genes are on the same chromosome or not. And at the end of class, I'm going to tell you a story, briefly, a genetic story, about why sometimes it's useful to know whether genes are on the same chromosome. And that'll wrap up genetic mapping for us today. So what's the next step? What do we have to do to figure out whether or not two genes are linked or unlinked? Pardon? Okay, so we can scratch out, we know that the tester individuals have to have gametes that have a little b with a little d. So in the offspring that are produced, thank you very much for noting that I'm disconnecting. So the F2 genotypes have to be combinations of the gametes that came from both of the F1 parents. Right? So it had to be one gamete comes from the F1 dihybrid, the other gamete comes from the F1 tester. We know the genotype of the F1 tester. Right? So we've got the little b and the little d in these F2 genotypes had to come from that parent. We've done this before. This is probably the third or fourth time I've reviewed this, so if you're snoozing, that's fine. You can go back in time and watch this later on a video. What's left then are the four different types of gametes that the F1 dihybrid produced. Independent assortment, if these genes are independently assorting, tells us we would get an equal ratio of big B's with big D's, big B's with little d's, little b's with big D's, and little b's with little d's. And that sets up our expectation for this ratio. That if it's true that the F1 dihybrid produces an equal number of those four types of gametes written up there in blue, then we should have exactly those ratios in blue, which should be 1 to 1 to 1 to 1, not 69 to 81 to 422 to 428. So which genotypes in the F2 of those four that are up here, which ones are there more of than we expect? When I say more of, that's the hint, which are the bigger numbers. Are the 422 and 428? This is the essence of genetic mapping. Are those the parental types or the recombinant types? Okay, so we know they're parental because, you're right, it's parental. We know they're parental because we look at the P0 parental generation gametes. Little b's with big D's and big B's with little d's. So those are parental type haplotypes. So of the total, this F1 individual, the F1 dihybrid, produced 1,000 gametes that we've looked at now, because we're looking at 1,000 of that individual's kids. So we're looking at fertilization events that involved 1,000 of the F1 parents' gametes. So we know that 422 of that individual's gametes were big B, little d. And 428 had a little b and a big D. What's the numerical pattern we're looking for if we want to know whether these genes are linked or not? If they're unlinked, what do we expect? If B and D are on separate chromosomes and independent assortment occurs, we expect to see that 1 to 1 to 1 to 1. We should see 250 of each of those 4 types. 250 big B, big D here, 250 of these, 250 of those, 250 of those. That would be a 1 to 1 to 1 to 1 ratio that totals 1,000, the same number of individuals we looked at. Then you would do a chi-square test to ask, is 250 different than 69? You do observed minus expected squared over expected. Is 81 different from 250? Is 422 different from 250? Is 428 different from 250, sorry. You add all those numbers up, and then you get the chi-square value. You look up the p-value on a chi-square table. Review upon review. So how many recombinants are there total in the F2 generation?
150.
69 plus 81. So we've got, right, big B and big D and little b and little d are recombinant types. Those are gametes that are not found produced by the P0 parents. They couldn't be because there was no big B and big D in either of the parents together. So we've got 69 plus 81 or 150 recombinants out of 1,000 total individuals, 69 plus 81 plus 422 plus 428. So what's the genetic distance in centimorgans between B and D?
15.
Okay, so which units would that be if you want to say 15?
Centimorgans.
Centimorgans or
Map units.
map units. They're identical. So recombination fraction in this case is 0.15. 150 divided by 1,000. When you multiply that by 100, you get map units or centimorgans. So the point of doing all of this is to help us address the question that we ended with last class. Here we have the genetic distance between genes B and D, 15 centimorgans. Try to remember that because I'm horrible with numbers and I can't remember that to the next slide. Any questions before we go to the next slide? Any at all? So, genetic mapping, dihybrid test cross, find the recombinant types, add them together, divide by the total number of individuals. Oh, crud, I've got to wait longer than one slide. Remember, there's how many centimorgans was that? 15. B to D, 15. Try to remember. So today is the last material that's going to be covered for the exam. So what we do on Monday, despite it being between us and the exam, will not be on the exam. So I trust that you'll show up on Monday anyway because it's interesting material and it will show up on the next exam. But Monday is kind of a freebie for now. I just sent out earlier a request to do exactly what we did in preparation for the first exam, for you to send questions that you've written that you think would be questions I would ask on the upcoming exam. I will assemble those into a study guide. So I'm asking you, please, if you're going to participate, optional. If you're going to participate, please send those to me by tomorrow night, even though I know you've got better things to do on a Saturday night. That gives me Sunday to put the guide together and get it to you, so you've got a couple of days to use it before the Wednesday review session. Wednesday review session will be like last time, bring questions, I answer them. Friday, exam. Any questions about scheduling? Concerns, questions, course mechanics. Otherwise, we get back to the genetics, I hope. Let's see what happens. Oh, good. Yeah, so we're still working on calculating genetic distances. Today's goals. Last class, we did these two examples. We calculated the distance from A to B. So the A to B genetic distance in this case was, let's see, we had 100 recombinants out of 1,000 individuals. So the A to B distance was 10 centimorgans. This is all review. I'm not going to show the details. So A to B was 10. We did this example in class last time, A to D. That was 50 out of 1,000 individuals having recombinant genotypes. So that's 5 centimorgan distance. So A to D is 5. And then at the end of class last time, I asked you to draw me a genetic maps that would explain how the distance between A to D being 5 centimorgans, right, which is true in all of these. So all 4 of these examples I'm showing that you submitted, you drew genetic maps where A and D were 5 centimorgans apart, and A and B were 10 centimorgans apart. And that's all consistent with the 2 pieces of genetic data we got last class. So there's A to B, 5 to 15 centimorgans, so 10 centimorgans distant. And the point here is that you can add genetic distances. So the sum of the distance between D and A, if it's 5 centimorgans, and if A to B is 10 centimorgans, then what should the distance from D to B be? This is the additive part, which we haven't covered yet. You can add genetic distances together, like physical distances. So what should the genetic distance between D and B be if this drawing is correct? If it's ADB, or sorry, DAB. Should be 5 from D to A, 10 from A to B. The prediction there would be the D to B interval is 15. But as this student drew, wait a second, why is D showing up on here twice?
It's showing both possibilities of where D could be.
So which is the right order? Is it DAB? Or is it ADB? So we've got 2 examples, 2 submissions up here. This one here in the upper left proposed ADB. Here in the bottom right was the same, ADB. And in the bottom here we have a different order of genes, DAB. Which is it? Both could work. In all of these examples, A and D are 5 centimorgans apart. And in all of the examples, A and B are 10 centimorgans apart. So where are we to put D?
Do we count the cross that we did at the beginning?
So now let's add in what we did at the start of class today. What was the A to, now I'd forgotten already. What was the A to D genetic distance? Or sorry, the D to B genetic distance. It was 15. So which of these options does that rule out?
ADB.
So it can't be ADB. If this distance, D to B, is supposed to be 15 centimorgans, then how would you get a 10 centimorgan calculation from A to D? So what this is saying, ADB, we've calculated that distance is 15, that distance is 5. Sorry, what's A to D? 5. What was A to B?
15.
I think A to B was 10, wasn't it?
Yeah.
Yeah. So that's not true. A to D 5, D to B 15, then the A to B distance should total 20. But we found it was 10. So the only order in which all three of these genetic distances make sense in this case is this answer. Because we just calculated that D to B was 15 centimorgans. And that's exactly what we'd expect if we added 5 centimorgans from D to A and then another 10 from A to B. So, summarizing. For any 3 genes, there are 2 possible, after you do 2 crosses, there are 2 possible orientations of the 3 genes, 2 possible orders. What do you have to do to know for sure which order is the right order? You do 2 crosses, you get the A to B distance, you get the B to D distance. You have to do the 3rd cross. There are 3 pairs of genes. If you're looking at the order of 3 genes, A, B, D, you have to look at the A to B distance, you have to look at the B to D distance, and then you have to do the 3rd cross, the A to D distance. And with all 3 of those pieces of information, clarity should emerge. So if you've got 3 genes, you have to do 3 crosses to be sure about the correct order of genes on a chromosome.
Would it matter if you would have put B, A, and B?
Yeah, that's a fabulous question. It comes up every semester and I always forget to add it. So I'm always glad when students ask. So is it bad? Bad. Is this different than what's drawn up here? Not really, because DNA is a linear molecule, and if we just, basically what we're doing here is we're just drawing this thing inverted 180 degrees. So the order of genes is no different. It's just relative to perspective. If I was standing on my head, then, which I would love to do, but I'm afraid something would fall down, then it would look like DAB. So the left to right order is not important. It's just the relative order, which is closer to B. Is A closer to B or is D closer to B? That's the question. So it's 3 genes with 2 crosses. You can make 2 different guesses about the order of genes. You have to have the information from the 3rd cross to know what true order is. And that's summarized on this slide. Does anybody have any questions about any of this? Now I'm going to ask you to apply this information. And maybe remember something from last class too. So here are physical maps, or genetic maps. We've got 3 different genes, Q, R, and T. And so I'm telling you, these are physical maps. R and T are linked. They're on the same chromosome. Q is on a different chromosome. So please, on this, if you have the technology at your command, draw what are the genetic distances between all of these pairs of genes, and take a screenshot, send it to this Google Classroom assignment, QRT genetic distances assignment. Feel free to talk to each other, it's not a quiz. Collaborate if you like. In about 3 minutes, we'll be coming back together and discussing.
If they're unlinked, they're going to be 50 centimorgans apart. And so the R and Q together.
Here's one submission. Recognize some of you are done talking, some of you are still talking. I apologize for those of you that are still debating. So here's one submission. What do you think? R and T less than or equal to 50 centimorgans? You agree or disagree? What is the possible range of genetic distances of two linked genes? What's the smallest possible genetic distance? Smallest possible. If two genes are right next to each other on the chromosome and they never get separated from each other by recombination, what's the smallest genetic distance you can have? 0. Okay, so if you see two linked genes, they're somewhere between 0 and what's the largest genetic distance you can have? 50. So R and T have to, if they're linked, be somewhere between, hopefully your answer is somewhere between 0 and 50. Can't be more than 50 because that doesn't happen. So independent assortment, Mendel's second law says that you expect 50% recombinants and 50% parental gametes, which equates to 50 centimorgans, half of the gametes being recombinant. So what's the genetic distance between genes that are on separate chromosomes? Those are the ones that are independently assorting. They're unlinked. So what's the distance between Q and R? 50 centimorgans. Now there's one pair that isn't diagrammed up here, but what's the other 50 centimorgan genetic distance for sure up here? Q to T, right. So there's 3 pairs of genes here. Q to T also being on separate chromosomes means they also ought to, we expect them to show 50 centimorgans, 50% recombination, 50% parental types. And of course, what everyone was discussing was what to do about R and T. And I wasn't looking, there's no specific answer. You can't tell from this drawing what the exact genetic distance is. I was looking like this for sort of a range. So how would you interpret that drawing? Less than or equal to 50 centimorgans? Does anybody have a different answer that they want to share? Because that's not necessarily the right answer.
It's less than 50, but not equal.
Right. So it's, yeah, exactly. It's less than 50, but not equal to 50, only in my opinion, because I drew this, so my opinion counts. The way I would draw this, if the genes were absolutely guaranteed to always have a recombination event happen between them, they would have to be at the absolute ends of the chromosomes. Because that's the only way to ensure that a crossover 100% guarantee crossover, XO. If the genes are absolutely at the physical ends of the chromosome, and we agreed, I told you to agree, sorry, about a week and a half ago, that one of the assumptions we were going to make is that one crossover always happens on every chromosome at every meiosis. So if there's a crossover that's going to happen on that chromosome, if R and T are here, as I drew them initially, it's possible that a recombination event could happen between that chromosome and sister chromatids during meiosis, somewhere outside of the R and T. So that occasionally the R and the T go together into the same gametes. But this was a reasonable interpretation of what I drew. The R and the T genes are really far apart. They're basically going to act like they're unlinked. Probably not going to be equal to 50 centimorgans, but they'll be close. So that's the upper range. The lower range, if this is a really bizarre organism, could be 0 centimorgans. So genes can be, in actuality, very far apart physically on a chromosome and still be very close together genetically. That's a detail for advanced genetics. We'll start talking about human genetics, maybe. So we might come back to that later in the term. So yeah, Q to R, 50 centimorgans, Q to T, 50 centimorgans, R to T, somewhere between 0 and 50, not including 50, in my opinion. Any questions? switch gears a little bit and talk about this concept called genetic hitchhiking and why it's sometimes dangerous to have genetic hitchhiking occur. I also call this when linkage goes bad. So there are sometimes it's useful to know about linkage. I sort of motivated this entire chapter at the beginning with the whole GFP roller worm story. Sometimes geneticists want to be able to combine traits from different organisms or different strains that they're working with to combine two useful traits into one organism. Think selective breeding. You want corn that makes lots of ears of corn, and each ear of corn has large kernels, so you want to breed those two traits together. There are some times when it's bad to have genes that go together. Linkage. So this is just one example. Crohn's disease, which is sort of a crude approximation. I don't suffer from Crohn's disease. I hope none of you do, but if you do, I hope you don't mind me talking about this disease. My understanding is, well, here, that's my understanding of Crohn's disease from this simple figure. It's related to irritable bowel syndrome. You have upset stomach, diarrhea, etc. And trying to figure out why Crohn's disease occurs and whether it's got a genetic basis, what causes it, has been an ongoing question just because it's so disruptive to normal life for the people that suffer from it. And geneticists doing genetic mapping identified a region of chromosome 5 in us, human chromosome 5, that seems to be associated with being susceptible to getting Crohn's disease. So geneticists pinpointed this particular region of chromosome 5. And what we're doing here is we're looking at, on the top here, this is an ideogram, right? The artistic drawing, rendering of chromosome 5. And underneath here are genes. So these are the positions of genes that are on our chromosome 5 and their relative locations. So we have the scale here. So chromosome 5, we have position 131,500,000, with nucleotide 1 being the far left end. So we're already 131 million base pairs into chromosome 5. To whatever here is 900,000 base pairs. So we're looking at 400,000 base pairs of chromosome 5. And that part of chromosome 5, just for scale, is what that red bar is on the ideogram of chromosome 5. So we've blown up that tiny little sliver of chromosome 5, and we're looking at what genes are located there. So geneticists think one of these genes, CSF2, P4HA2AS, geneticists are not necessarily clever with naming genes, PDLIM4, SLC22A5, SLC22A4, IRF1, IL5, the names of some of the genes that are in this relatively small part of chromosome 5. So our goal is we're going to figure out, well, we're going to pretend, we're going to go on a quest. We're going to go on a quest to find, to find which of these genes is responsible for Crohn's disease. So here's a little bit of information, then I'm going to ask you to make a prediction. I'm just going to pick on a few of these genes. So OCTN1, which is also the gene that's called SLC22A4 in this map, is a dietary antioxidant transporter. So it's a gene that encodes a protein that gets expressed in our guts, literally in the intestine, that takes antioxidants, those good free radical fighting things that you're supposed to eat the rainbow of fruits and vegetables and drink red wine and all that to get antioxidants to combat the free radicals. So this is a gene that does that. It's expressed in our guts. It pulls those antioxidants from our diet, from the food we eat, into our bodies for use as antioxidants. We've got interferon regulatory factor. Has anybody taken immunology here yet? It's an immune system related gene, interferon regulatory factor. And we've got an interleukin, which is another related gene. So if you go to Socrative, please just vote anonymously if you want. You don't have to put your name in if you don't want to. Which of these genes, just based on what I've told you, do you think is responsible for Crohn's disease? Mutation in which of these genes? So just to help you translate, OCTN1 is the one that's also known as SLC22A4. That's IL5. This is IRF1. So you are split between SLC22A4 and IRF1 in terms of which one probably causes Crohn's disease. So it turns out, this is not the answer, this is just part of the story, that there's a mutation in people that have Crohn's disease, correlation. A lot of the people that have Crohn's disease have a mutation in their OCTN1 gene. Bad or good? Are mutations always bad? What's an example of a good mutation? What's a mutation that made a phenotype that's useful for humans? Mutation that allowed us to see 3 different colors, that allowed the evolution of trichromacy, the ability to see 3 different colors, red, green, and blue. Other types of mutations that are useful for humans? What about the mutation that causes sickle cell anemia? Useful in some populations where, if you've heard this story before, it's bad if you're homozygous, but if you're heterozygous, it protects you from malaria. So that's an example of, depending on which part of the country, of the world you live in, sometimes it's useful, sometimes it's not. This is a similar story. This genetic hitchhiking phenomenon. Is a mutation that increases dietary antioxidant absorbance going to be better or worse for you? What do you think? Is it good to have antioxidants taken up from your diet or not? Good? Bad? Okay, so most of you are saying good, which is true, at least that's what biologists think, that this is a beneficial mutation. That it's useful to the people that have it. It helps them bring more antioxidants from their diet into their bloodstream to help fight the free radicals, especially reactive oxygen species produced during electron transport, production of ATP by mitochondria, and that whole story. So that was one thing that scientists knew going into trying to figure out what was going on with the genetic basis of Crohn's disease. So then, sorry, you can't see the citation down there at the bottom of the screen, Huff et al. So 2011, this team of geneticists wanted to figure out which of those genes from this region causes Crohn's disease. So we're looking at, I'm just going to draw the connections between the genes that they're looking at. So I'm showing you a graph from their manuscript, data, that's the bottom half, and the top half is this map of the genes in the region that we're used to looking at. So there are the 6 genes that they studied and the relative positions along chromosome 5. What would your prediction be about, and how did geneticists go about figuring out what genes cause what traits? The easiest thing to do, and that's what these scientists have done, is they've taken DNA samples from people that have Crohn's disease and the ones that don't. That's what these categories Crohn and control are. So they've got DNA samples from people that have the disease and don't. Ones that don't are the control participants. What do you think they're going to look for? What would you predict about genetic differences between these 2 classes? If you compare individuals that do have Crohn's disease and don't, what are we going to look for? So what sort of DNA sequence differences?
Production of something else. Someone with Crohn's versus someone who doesn't have Crohn's.
So you're looking for any sort of difference, right? I mean, that's why we categorize these people. So this is how we look for the genetic basis of diseases. You take people that have the disease, people that don't have the disease, and you compare them genetically. Find out what's different about them. These scientists had the idea, although now this is really not good because you can't see that axis. There you go. On the left side, the y-axis, relative messenger RNA expression. So what they're measuring here for individuals that have Crohn's disease and individuals that don't, for each one of these genes, right? So for each one of these genes across this region that contains a gene that we think causes Crohn's disease, they've done the same thing. Compare control and Crohn's disease patients, and they want to know basically how much of that gene is produced. That is how much, the messenger RNA, of course, is transcript. You know this from knowing about the central dogma. So they're not measuring the amount of protein that's produced from each of these genes. They're just measuring how much messenger RNA is produced from these genes, which might have something to do with how much protein is produced from each of these genes. And their hypothesis is, if there's a gene here that controls, that regulates whether or not you get Crohn's disease, you should see what? When you look at the gene expression, the amount of gene produced, transcript produced, what would you expect to see comparing Crohn's versus control?
More transcripts.
More transcripts in which category? Would you expect, for any gene that controls Crohn's disease, would you expect more transcript in Crohn's disease or less transcript in Crohn's disease? For the first time ever, I'm happy to tell you that your silence paid off because you're all right. So what question would you want to ask? There is no answer. There is no right answer to that question. You can't tell. There's no way to know. There's no way to predict whether or not you would expect a gene that causes Crohn's disease to be more highly expressed or less expressed. Depends on what these genes do. So what were they looking for if they didn't know which direction the gene expression should change depending on whether or not you've got the disease? What do you look for? Just a difference. They don't know which direction. They don't know if they're going to look for a gene that's higher in Crohn's and lower in control, or higher in control and lower in Crohn's. They just look for a difference. Where do we see the differences up here? In each of these cases, basically, they're doing this. They're just looking at the comparison between the control and the Crohn's. Is there a difference in the amount of messenger RNA? What about this case on the left? P4HA2. Different?
No.
No. Okay, where's there a difference?
Octen 2.
So there's a difference here in octen 2, it seems like. I mean, just looking by eye. Good first pass. Where else? IRF1, there's also a difference. And they're in the opposite directions. So again, reinforcing the idea that we couldn't predict in advance whether or not changes up or down would cause Crohn's disease. Where on here do we see something that gives us more information about whether or not these differences are real? We've got statistics. So in this case, they actually statistically compared the differences between control and Crohn's. Which gene is most significantly different between control and Crohn's patients? It's the one with the smallest p-value, you're right. It's IRF1, with a staggeringly small p-value of 0.0036. So there's the biggest statistically significant difference here. The people with control have relatively low expression of this gene. People with Crohn's have elevated amount of transcription and then presumably protein production of the IRF1 gene. So the conclusion of the paper is that it's IRF1 that's responsible for Crohn's disease. That there's something different, maybe there's a mutation, there's something that increases expression, increases transcription from that gene in people that tend to get Crohn's disease. That doesn't prove that this gene causes Crohn's disease. This is a correlation. People that have Crohn's disease tend to have more IRF1 messenger RNA. But it's a pretty good start. So they can do other studies to ask whether or not it's IRF1 that actually causes Crohn's disease. So here's where we finally get to hitchhiking, and then we're done for the weekend. Woo! Except I have lost my connection. But remember, where was that beneficial mutation?